research reel with Ashley Eadie. I'm so excited to be able to present her today. So just a little bit to start off about who is IMPART, just to kind of share with anyone who's new joining us here today. So IMPART Investigator Team is a national virtual research institute headquartered at Dalhousie Medicine here in St. John. And our team is composed of 45 academic and clinical partners across 8 universities and 6 different provinces, as well as 25 community and funding agency members and 15 private sector companies and 2 provincial government departments. IMPART's mission is to think boldly and utilize our network to improve the outcomes of vulnerable patient populations, starting always with innovation developed and tested here in New Brunswick. We apply intersectional thinking between social and medical innovation that have brought together a diverse and inclusive team of experts to support activities from fundamental research to applied interventions with vulnerable populations. So that's a little bit about research going on in IMPART and how the team works here at IMPART, but a little bit more about research reels that we're presenting today. The research reels webinar series is hosted monthly here within the IMPART team. And we typically highlight investigators or trainees that have ongoing or completed research within our network. The objective of this webinar series is to improve the networking amongst the IMPART team members, enhance opportunities for future collaborations, and support knowledge translation for our research findings here at IMPART. So just a little heads up for today in particular, this research reel will be recorded and posted to YouTube. So for anyone watching later, thank you so much for joining us. And for anyone who didn't know, feel free to check out those recordings on our YouTube page. Without further ado, I'll introduce today's speaker. So welcome, Ashley Eadie. I'm so happy that I am able to introduce her today because she has helped me a lot along my way on my project. So without further ado, Miss Ashley Eadie. She is a 6-year PhD candidate in the Faculty of Medicine at Dalhousie Medicine New Brunswick here in St. John, and her research focuses on the underlying mechanisms of heart failure in order to better study identify effective treatment strategies, as well as Ashley completed her BSc with honors in biology and molecular toxicology at the University of New Brunswick in 2014, and subsequently defended her MSc in pharmacology at Dalhousie in 2016. During this time, Ashley also served as an engineer on board the Kingston-class coastal defense vessel as a member of the Canadian Armed Forces, and was awarded the Queen Elizabeth II's Diamond Jubilee Medal for outstanding service in Canada in 2012. So congratulations, that's a huge accomplishment. And Ashley's research has been published in top-tier biomedical journals, including Science Translation of Medicine, the American Journal of Pharmacology, and the Journal of Molecular and Cellular Cardiology. And her work is supported by funding from the New Brunswick Innovation Foundation, Heart and Stroke, and the Nova Scotia Health Research Foundation. So without further ado, I pass it off to Ashley for her presentation today with an interesting take on FDA regulations.
Thank you very much, Tori, for such a nice introduction. I am really excited to be here today. Let's just get those slides up and going. What do we see?
Just 1 second. It looks like it's trying to share, but I can't see the whole screen yet.
Yep, we'll just give it a second. It's a bit. There we go.
Awesome. You're all set to go.
Perfect, perfect. What's a presentation without a little bit of technical difficulties? So as Tori mentioned before, I'm a PhD candidate in the Department of Pharmacology. And today I'm going to be presenting some of the work that's a little different from my thesis about insights from public regulatory databases. So if you're not familiar with pharmacology and what pharmacologists like myself do, we study drugs, how drugs affect the body, which is pharmacodynamics, and how the body in turn affects drugs, pharmacokinetics. And ultimately, we take that knowledge and try to synthesize new ways to improve patient outcomes. A particular branch of pharmacology that I'm interested in is that of translational pharmacology, which is trying to take a broader consideration of how drugs move from the lab bench to the patient bedside. One of the challenges that we face as pharmacologists is that of evidence synthesis. So trying to figure out where all of the background information that has been published over the years in the primary medical literature is, and how we can cultivate that in a way that's efficient, so that we can build better drugs from learning from the past. But a lot of that background information is scattered across journals, across generations, across different author groups. Not only that, but there is incidences of reporting bias noted in the primary medical literature. So studies, for example, with negative results are less likely to be published. So as a result, these more traditional resources often are an incomplete reflection of all drug information. Any negative findings, adverse events that could be of use and interest to research like myself, prescribers in medicine, as well as patients in the general public. So to overcome these challenges, we've been looking towards regulatory agency databases, particularly the one that I'm going to talk today about pertains to the FDA. So regulatory agencies like the United States Food and Drug Administration are responsible for reviewing evidence of safety and efficacy for drugs prior to their market approval. So that means that they take all the clinical trials and the preclinical trials and pharmacological trials and safety and efficacy studies. Those get submitted by the drug sponsor or drug manufacturer to the regulatory agency for regulatory review. Following regulatory review, those drugs either get a rejection or market approval. But in those regulatory reviews, we can gain a lot of information that's not necessarily available in the primary literature. So drug strengths, weaknesses, and opportunities for better drug development. The regulatory agency databases are an often overlooked source of valuable pharmacological information for the medical community. It often includes information excluded from the primary literature. To highlight how we can gain new insights through full drug approvals, myself and Professor Matthew Herter from the School of Law at Dalhousie University, along with Dr. Keith Brunt, and the help of Leslie Ogilvie at the University of Guelph, explored how the Food and Drug Administration reviewed and approved the new drug Entresto, also known as sacubitril and valsartan, highlighting the value of these FDAs through this case study. Entresto is a drug developed by Novartis for the treatment of heart failure with reduced ejection fraction in adults. It's a combination of two entities, sacubitril and valsartan, and together these make up the first-in-class drug known as Entresto. That's why it's also particularly interesting for this case study as a first-in-class. And to this date, it remains the only in-class angiotensin receptor blocker neprilysin inhibitor. So in 2015, it was found that Entresto was very successful in treating heart failure with reduced ejection fractions in adults, reducing cardiovascular death due to heart failure, reducing cardiovascular death or heart failure hospitalizations compared to the existing standard of care. So it was granted approval by the FDA in 2015. A subsequent trial in 2019 looked at Entresto's use for the treatment of a different kind of heart failure known as heart failure with preserved ejection fraction. Although heart failure with reduced and that with preserved ejection fraction differ in many aspects, the underlying mechanisms that push that cardiovascular phenotype towards heart failure, such as fibrosis and hypertension, are very similar. And so when it failed to reduce mortality and hospitalizations, it was quite a surprise based on that expectation that those mechanisms were similar enough to be treated. So our goal here was to explore what knowledge gaps we could from the original reduced ejection fraction application for Entresto, and use that to see what may have contributed to the failure of preserved ejection fraction treatment with Entresto later on, and figure out what those knowledge gaps might mean for future clinical populations and opportunities to develop better drugs in the future. So a bit of a spoiler, we found 3 principal findings when we went through the regulatory agency databases. First, pertaining to sex differences relating to heart failure etiologies between reduced ejection fraction and preserved ejection fraction heart failure. The second, pertaining to disagreements with the mechanism of action between FDA reviewers relating to its combination of 2 drugs and what each component had to play as a role. And the third, pertaining to pediatric heart failure and the use of biomarkers in a follow-up study or follow-up submission, and how that related in turn to ongoing adult post-marketing requirements issued by the FDA for safety. For the purpose of today's presentation, I'm just going to focus on the first knowledge gap that we identified. So one of the salient features that differs between reduced ejection fraction and preserved ejection fraction heart failure is that preserved ejection fraction has a higher incidence in females. So keeping that in mind, what we did was we went into the publicly available regulatory approval documents from the Drugs at FDA website and retrieve those documents pertaining to Entresto. So using that search term Entresto as an opener, we went through and collected approval dates, letters, correspondences, labels, memorandums, and reviews linked to Entresto's original reduced ejection fraction application, and went through those. So one of the benefits of going through the Drugs at FDA database particularly is that they make their FDA reviewer reviews public. And so you can gain more information as well from the rationalizations that they put into their arguments and their conclusions. So from those documents, we went to, we specifically focused our interest towards the pharmacology review. And in the first section here, pertaining to studies reviewed and studies not reviewed. So studies that were submitted to the FDA and where they were and what their data was interpreted as. So what we initially found was that although a higher proportion of females was included in the preserved ejection fraction trial, where we have a higher incidence of females in that heart failure etiology, the pharmacokinetic studies that were used in the original application were studied largely in males. So what we saw was essentially a bias in female recruitment. And I'll give a couple examples of that here. So in the first example, females were not included at all in Novartis's thorough QT study. So the QT study or thorough QT study is an FDA required study to assess whether a new drug causes a potentially fatal condition known as arrhythmia, in particular a form of arrhythmia known as torsade de pointe. In its QT study, Novartis included 81 males and no females in that study. I should mention also that the incidence of arrhythmia is observed to be about equal between males and females. It's not something that occurs only in males. There's equal susceptibility. The second example that we saw of this unconscious bias in female recruitment is that females were observed in some of the studies cited within the FDA's documents as they were recruited rather under stricter criteria than males. So in this study cited within the FDA's documents and within its label actually, published in 2010, both studies, the methodology cited both studies enrolled healthy male and female volunteers aged 18 to 55 years However, female volunteers had to be postmenopausal, so no hormone replacement therapy in the past 6 months, or had to have overgone ovarectomy with or without a hysterectomy for inclusion in this trial. And so understandably, this trial only wound up recruiting 1 female to 48 males. So the same strictness of criteria for males was not there. There was no mention of testosterone therapy or gonadectomy for inclusion in them. We also found that the studies that did include females were not separated by age or sex. So essentially we saw a phenomenon known as data pooling, which makes it difficult to differentiate sex-specific differences between males and females in pharmacokinetics behind age and sex-dependent differences. So this is another one of the studies that was cited within the FDA label, looking at sacubitril levels in the plasma over time between females and males. So age category was very broad, in that it included anybody from 18 older. So it makes it very difficult in this sense to differentiate what age contributes and what sex contributes. So sex, especially because sex-independent data within the same study demonstrated that Entresto clearance was significantly reduced with age, indicating that there could be a masking effect here. So to summarize, increasing the number of females does not necessarily mean optimization of a clinical trial. Just powering up with more females in your study does not help, essentially. Lack of female inclusion could explain part of the Entresto's failure to confer benefit in that sense, the same sense as reduced ejection fraction as that with heart failure with preserved ejection fraction. Studies like these also present opportunities for further sex-dependent research. So, for example, what could be done right now is to collect market data from females that are already being prescribed Entresto and deduce whether that change in pharmacokinetics is present, and if it could compound with regular use of Entresto over the long period of time in which somebody would have heart failure. And it also highlights potential systematic gaps in FDA reviews. At no point in time through the thorough QT study or anywhere else within the FDA documentation was female disinclusion or female unconscious bias in recruitment mentioned. So we might have a larger issue on hand that this type of study can help identify. But what I hope you can take home from this particular presentation is that the regulatory agencies' documents, like those with Health Canada or the European Medical Agency or the FDA, those type of documents and databases hold really valuable insight into drug strengths, policy strengths and weaknesses, and that they provide really important lessons for improving novel therapeutics going forward. How we design clinical trials and preclinical trials, how we design policy, and how we improve patient outcomes. If you'd like to learn more about that, you can always just visit the Drugs at FDA Approved Drugs website, or check out our publication below from our group that gives a further breakdown, a more extensive breakdown of how we actually go about doing that and how you can replicate studies like that on your own. And with that, I will turn this presentation back over to Tori for questions.